hey, we need to know what the cutoff point is, okay? Because the exam is Friday, and some of the cell structure will be on there, okay? But not everything. All right, so I think we left off here on Friday looking at this idea that when you look at the phospholipid bilayer and how the glycerol and the R, by the way, are the, like for the bacteria, the R bacteria, the R letter designates fatty acids. And so that's what we were looking at, you know, here. The fatty acids coming off the phospholipid, and you have the fatty acid chains coming off of that. And that's what those are trying to depict. And then I said that the ether versus ester bond is a big deal, even though it sounds like a picky detail, but that ether linkage for the archaea is something that provides some extra strength for the cell membrane of the domain archaea. And so them having that ether link does help. So that's the big difference between the archaea type and the domain bacteria and eukaryotypes. Another big difference is that the R groups here in the domain archaea are actually isoprene units. Isoprene, not fatty acid tails, but isoprene tails, okay? And so this figure C on the far right, that's the basic unit of an isoprene. They're 5 carbon molecules with a little methyl offshoot like that, okay, a branch. And so the R in the domain archaea are just long strands of isoprene units strung together. Okay, so that's another big difference. And that also can help them in more hostile environments to maintain a stable cell membrane. Okay, so the cell membranes can differ. Let's look at what that might look like. So here you're seeing the ether link up top. So again, if it's an ether link, that means it's the archaea. And these are the isoprene units hanging off of that, okay? So sometimes, I wish the artist had done this a little differently. So this can form a bilayer where this figure A, you can just take this and flip it and create space in between here, then it's a bilayer situation. Okay, so just take this image and flip it, leave a space, and then you have a bilayer membrane that particular archaea would have. On the other hand, figure B, that is not a bilayer configuration. This is a monolayer configuration. So you don't see a gap anywhere. It's just solid. So where we say, you know, like we have phospholipid bilayers. Here's another depiction of it. So here's figure A is a bilayer because you have here image with a gap. But the monolayer idea is that those isoprene units just go solid all the way through. That make sense? Yes or no? Okay. So that also is an advantage under certain conditions. So monolayer like that can be very advantageous if you have a solid string of isoprene units all the way across. It does help, especially under high pressure, high heat. It helps the microbe withstand pressure and heat pretty nicely. Whereas if it's a bilayer with a gap in between, those cell membranes can fall apart under high pressure and high heat. So archaea have some little extra design tricks to deal with some of the more challenging environments their faceplate. You can also see here that there's like ring-like structures sometimes that will form. So a little bit more complicated than what you see with the domain bacteria and the eukarya. Okay. So cell membrane structure can vary, even though, you tend to get taught just that there's only one type of cell membrane out there. Yeah, maybe with the animals, but when you get into the microbial world, it can vary. Pretty much any cell membrane, though, you need to think about what is it doing for the cell. It's a permeability barrier. It's letting certain things in and out, other things don't get access. So it's semi-permeable. And certainly that holds true in the microbial world. You see in the middle depiction there, they're showing that you can stick a bunch of proteins in it, and that can be handy. It's a handy spot to kind of set up your proteins and allow them to do their job, primarily with transport for us. With microbes, though, most microbes don't have any kind of, so in BI 111, you're probably taught prokaryotic cell types, which, the domain bacteria, the domain archaea, they're prokaryotic. They don't have organelles. That's what you get taught. And for the most part, that's true. But I will tell you there's a couple of exceptions. But let's just say that. Okay, prokaryotic cells do not have organelles. That means they don't have internal mitochondria. They don't have chloroplasts in there. So like right now, if you had lunch, your mitochondria floating around in your cells is busy making a lot of ATP for you. Bacteria don't have a mitochondria to do that. So they devote their cell membrane to that, as well as being a semi-permeable barrier, protein anchor. And so the cell membrane of microbes, the bacteria and the archaea, really are incredibly important because they stick a lot of the other functions that we do, like in our mitochondria and or in our endoplasmic reticulum or whatever. All right, so they have to stick the proteins needed for making ATP. They put that in their cell membrane. So it's an extra thing that they have to deal with. The cell membrane is really a workhorse for the microbe. And so we'll get into that as we get into metabolism, how they're using their cell membrane as part of their physiological processes. Okay, so all microbes have a cell membrane. Almost all microbes have a cell wall as well, but not all. So if you've ever had walking pneumonia, if your doctor said, oh, you have walking pneumonia, those are caused, that disease is caused by mycoplasmas, and they're famous for not having cell wall. And so when I had walking pneumonia, the doctor said, oh, you have pneumonia. He didn't say walking pneumonia. He just said, you have pneumonia. And he gave me a bunch of penicillin, which back, I don't know if they're still like that, but they looked like horse pills. They were huge. Ugh. And so I took all the pills, and a week later, no better. I go back, oh, you probably have walking pneumonia. So then he gave me a different set of pills. The penicillin made no difference because penicillin attacks cell wall synthesis. And I was sick with something that had no cell wall. So pretty annoying, but really graphic example of the idea that some microbes have cell walls and others do not. And so all the microbes that you will work with in lab have cell walls. I don't work with the mycoplasmas because they tend to be pathogens. So we don't work with those. So here you're seeing a very simple depiction of the difference between a so-called gram-positive bacteria and gram-negative bacteria. So we're looking at their, your book often calls them the cell envelopes. I learned it as cell wall, even though here you look at a gram-negative, it's the so-called outer membrane. So yes, it's the membrane, but it's actually part of the cell wall, or your book says it's part of the cell envelope. So, but I tend to say wall, so just be aware of that. So here you can see the brown. You compare the brown, that's the peptidoglycan. So in a gram positive bacteria, you see it's really thick layer of peptidoglycan. In a gram negative bacteria, it's very thin. That's a huge difference that you need to key in on. That will make a world of difference when you do your gram stains tomorrow in lab. That difference in thickness is the main reason why the gram stain works so well in the lab and will allow you to pick apart, oh yeah, this is gram positive, this one's gram. Okay, and then the other thing you can see is that outer membrane with the gram negative cell type. So 90% of a gram positive has 90% of their cell wall is peptidoglycan, only 10% with a gram negative cell type. So what are we talking about? The cell wall of gram positives and gram negatives being depicted here. And I'm going to go through some of this in more detail. Again, the kind of the blue and the purple here, hexagonal shape, that's the peptidoglycan. And it's very thick. The left, very thin on the image on the right. So here, oh, and then you have the electron microscope pictures up above showing the difference. When you look at it under the electron microscope, here's a good rendition of the top. All that big purple swath is peptidoglycan. Down here with the gram negative, it's very thin purple, okay, peptidoglycan. And then you're going to do the acid fast stain on Thursday in lab, and those bacteria have some additional things in their cell wall that makes them unique, and you have to do a special acid fast stain in order to easily see them under the microscope, which you'll learn that technique. So with a gram negative bacterium, you have, I like this image because they're showing just how busy a bacterial cell is, all kinds of stuff packed in there. And then you have that outer membrane and that thin peptidoglycan. We're even showing a flagella on here. All right, so they've done quite a bit of microscope work looking at the difference between the cell walls of gram negatives and gram positives, and they've come down to a couple of things to think about. So peptidoglycan, when you look at its chemical nature, is basically sugar bonded together in a very particular pattern. And so there are sugar derivatives, technically. So one sugar derivative is N-acetylglucosamine, and the other one is N-acetylmuramic acid, which are both challenging to keep saying that. So we abbreviate down to NAG and NAM. Okay, NAG and NAM. And so if I ask you an essay question and you could just put NAG and NAM instead of spelling it all out. Okay. Okay, so a good chunk of peptidoglycan are these sugar derivatives. Then we also have some amino acids in the mix. So there's certain amino acids, L-alanine, D-glutamic acid, diaminopimilic acid, which is abbreviated down to DAP, which you can abbreviate as well. In gram negatives, we see DAP. In gram positives, they replace DAP with L-lysine. Seems pretty consistent. So gram positives tend not to have that DAP. They put L-lysine in place instead. And then the last amino acid is D-alanine. So you see there's 4 amino acids, and they will be bonded together. I'll show you the overall configuration. So L-alanine, D-glutamic acid, DAP or lysine, and then D-alanine. So these sugar derivatives and these amino acids, they often say, oh, they form a glycan tetrapeptide. Tetrapeptide is referring to the idea that there's 4 amino acids. So when you string together just a few amino acids, we start saying it's a peptide. So tetra is 4. So glycan is referring to the sugar derivatives. So glycan, NAG, NAM, tetrapeptide, those 4 amino acids. Okay. Then those get put together in repeating units and bonded together. So let's look at that. So here they color coded the peptidoglycan, the blue and purple. And so you can see they alternate in long chains, they're strung together in long chains. Does it say which one is, oh, here, G for nag, the purple is nam. So it's nag, nam, nag, nam, nag, nam, nag, nam, long chains. And then you can see right where the nam is located, wherever there's a nam sugar derivative, you see those little colored balls? Those are the 4 amino acids. So you have these long chains of sugar derivatives, like they're like laid down right next to each other, but you have to have a way to keep them together. So those 4 amino acids will hook into the NAM, each chain, to keep them from flying apart. Okay? So here they've blown up, they've taken the NAM and then the 4 amino acids, and then the NAM next to it. And so you can see the 4 amino acids coming off of this end, NAM, bonds with the 4 amino acids coming off the neighbor chain. So that's what they're showing in this big blow up here, are the NAM. So here's the 4 amino acids coming off of one chain, bonding here. They're showing the bonding down to the next 4 amino acids in the neighboring chain. And they both are coming off of the NAM molecule. So with that configuration, they often refer to this overall configuration where you have these long sugar chains next to each other and then the amino acids hooking into the neighbors like that. And then if you look at the overall pattern, they often say it looks like chainmail, like the knights in armor, they wore the chainmail. So they often will use that word. They'll say it looks like chainmail. I don't know if that's relevant. Do you know what I'm talking about when I say chainmail? I don't know if that's super relevant, but if that helps you to think about its overall like three-dimensional pattern of how it gets put together. Okay, so with gram positives, they have many, many, many of these layers of the sugars and amino acids that build up around the cell membrane. Oh, let me show you this slide. This one I wanted to put in here to show you how the NAG and NAM are bonding together. So they go nag, nam, nag, nam, nag, nam, and they're put together with a beta-1,4 bond in between the two sugars. And then you see here, lysozyme sensitive bond. So if you have the beta-1,4 linkage between NAG and NAM, this enzyme called lysozyme can attack it and break it. So that's good. You have lysozyme in your mouth and in your tears. So when you eat food, for example, or drink water that's contaminated with a microbe, your lysozyme immediately starts acting on the beta-1,4s. And so right away, your body is trying to defend you and protect you. If enough of those get broken down, the cell will lyse and break open and will hopefully bother you. And so not all, so like the domain archaea, they don't have a beta-1,4 bond. And so your lysozyme wouldn't really do anything. But that's okay because we don't know of any archaea that hurt you. There's no pathogens that fall into the domain archaea. So we're good there. Okay, so in words, what we were just looking at, they say the NAG-NAM chains, they're like a sheet-like, they're laid down in these big sheet-like configurations where you have the peptidoglycan, running parallel to each other. And then you have those amino acids cross-linking. The fancy word is cross-linking the chains. And in those figures, you refer back to them, they've denoted like, okay, you see DAC in the red negative, DAC will hook up with the D-alanine in the neighbor peptide chain and the L-lysine to the D-alanine. Depends on if it's a gram negative or a gram positive. Now, they've discovered that gram-positive has some extra stuff in this region. And so they'll call them peptide interbridges. They have interbridges, and I'll show you a picture of that. So it's a little bit extra amino acids that get stuck in there. I'll show you. So again, maybe you're just going to have one sheet of peptidoglycan, like gram-negatives tend to have just that, but some like gram-positives, they have multiple layers, multiple sheets stacked up. And so this peptidoglycan, it's not really a huge barrier in the sense that like molecules can come through the cell wall area. It's really the cell membrane that does the job of really being picky about what comes across. So the cell wall is rather open in that regard, like chemicals can come through there. But it does give rigidity and structure to the cell so that they won't explode under conditions that might cause that if they didn't have cell wall. Okay, here's the interbridge idea. So here they're showing they studied Staph aureus, which is gram-positive. So they're showing the 4 amino acids coming off of DAM in the sugar chain, and then you see that there's this interbridge of glycine that it connects to, and then that interbridge connects to the next 4 amino acids in the neighboring chain. So sometimes you'll see that kind of extra, embellishment with gram-positive. So, and it tends to be glycine. They tend to stick glycine, which is a short amino acid. So sometimes you'll see that. And then the chain mail idea. Often they try to depict that sheet-like configuration that kind of looks like chain mail. So when you do your gram stain tomorrow, you're looking at the differences in the thickness of the peptidoglycan is really what's causing that gram stain reaction to work out the way it does. And then the other thing that gram positives tend to have quite a bit of are these called teichoic acid. And teichoic acid can be, here you see like plain teichoic acids here, and then you see lipoteichoic acids. So really, if it's teichoic acid, it's ordered, it just kind of sticks in the peptide. Lipoteichoic is going to further down fatty acid region. That's the only difference there. So it can vary in their overall size, but gram positives tend to have quite a bit of teichoic acids mixed into their peptidoglycan. And again, they think that it does give some structure and rigidity. And they are negatively charged. So it gives an overall negative charge to the cell surface, which is important when you think about different stains. If the stain is negatively charged, it won't cover a negatively charged cell. So you have to know your stains. What stains tend to have either positive or negative charge. So when you work Thursday, you're going to do a negative stain technique, and the negative stain is negatively charged. And so it's going to not make your cells stain. It's going to stay in the background, and your cells will stay unstained. It's kind of a weird look, but cool. Okay, so gram positives, fairly straightforward. Really thick peptidoglycan, acids in there, and that's pretty much it. Gram negative is a little bit more complicated, right? They are showing an extra part. They have this outer membrane that is on the outermost portion of the cell. All right, so this outer membrane, it's not like the cell membrane. There's some key differences. And it's often referred to as a lipopolysaccharide membrane. And I'll tell you why. So if you've had MedMicro, they might have talked to you about LPS. That's short for lipopolysaccharide, the LPS. And so it's important when you think about how pathogens might affect us. Often the LPS is what's making us sick. And I'll explain a little bit more about that. You have this outer membrane. then you have a space, and they have to give that a name too, so they call it the periplasm. That's just the physical space between the outer membrane and the cell membrane, essentially. In the periplasm region, you'll have the peptidoglycan. So you'll see the peptidoglycan hanging out in that space, the periplasm. Thin. And then after that, you'll hit the cell membrane after the peptidoglycan. So that outer membrane, again, not very selective on what comes through it. So it can, you know, omit some bigger molecules that just physically can't fit through, but it's not as picky as the cell membrane. It's not that level of semi-permeability. Okay, so this is kind of a mock-up of what it looks like for the cell wall of a gram-negative. So the purple, you can see that they've color-coded purple and then the pink. And then you get into this region. Well, all right, let me start. I like to start with the very, very outermost region. So you see the purple here? Let's start with that. So let me go into the next slide. The purple structures we were just looking at, those are called the O-specific polysaccharides. They're just sugars, special sugars. You don't need to know that they're hexyl sugars and dideoxy, but sometimes people have asked. Just know they're polysaccharides, okay? Then, so those are the purple things we were just looking at. Then you can see they're bonded to the pink stuff next, right? So purple to pink. The pink region, that's called your core polysaccharide region. And again, if you're curious, it's composed of a variety of different sugars. You don't need to know these names. Just know that I want you to know the word or phrase O-specific is the most outermost polysaccharide, then that's bonded to the core. And then that yellow thing, I'll go back, is called the lipid A. So let me go back to the image. All those yellow squares, those are lipid A molecules. Okay, lipid A, that's what our body can overreact to, the lipid A portion of the lipopolysaccharide. So what I just described to you is the lipopolysaccharide. The O polysaccharide, the core polysaccharide, the linker, and then the lipid A. All of that is called the lipopolysaccharide. Lipopolysaccharide. And so gram-negative microbes have that going on as part of their cell wall or cell envelope. And it's really the lipid A. If this cell breaks open, lyses and breaks open, and that lipid A gets free, so our body can come into contact with it, our immune system tends to really go wild. And if it's a big enough all at once release of lipid A, you can go into septic shock and die. And so if you're sick with a gram-negative and you're taking antibiotics, you need to make sure, your doctor needs to make sure that that's a safe thing to do, because you don't want millions of gram-negatives lysing in you all at once, because then your immune system might go kind of nuts. So some consequences there. So they refer to the lipid A, that yellow square. They say it's an endotoxin to our bodies. It acts like an endotoxin. And I'll get into exotoxins and endotoxins. We'll get into that a bit later. But definitely our body reacts to it very strongly. Okay, so then the lipid A are the yellow squares, and then you can see there's some things dangling off the yellow squares. They're trying to detect fatty acids dangling on the yellow. So lipid A has fatty acids attached to it. Okay, so that's the last part. So this whole slide, whoops, that's describing the lipopolysaccharide. Okay? And it's just the outer half or the outer leaf of the outer membrane. That's what we refer to as the lipopolysaccharide. So let me go back to that image. So the outer of the outer, this whole thing, so I'm talking about at the end of the fatty acid, they coming up. That's the outer leaf of the outer. It's quite complicated. The inner leaf is this section here. The inner leaf of the outer membrane, it looks pretty normal. It just looks like a kind of a typical phospholipid layer there. And that's pretty much what we see. All right, so it's that outer half of the outer membrane that's really complicated. So let me go back. Oh, here they've color coded it again. So the purple is the O region, and then the pink is the core region. And then you have the lipid A with the fatty acids dangling off of it. So the inner leaf is pretty straightforward. It's just some phospholipids. You have some proteins stuck in there as well, lipoproteins, and they help attach the outer membrane to the glycan so it doesn't like fall apart. So you have some anchors. The lipoproteins kind of serve as an anchor. And then you have these special proteins called porin proteins. These are channel proteins. They form a channel in the middle. And they span the entire width. And they allow primarily hydrophilic molecules, so water-loving molecules, to come through, and small. Low molecular weight just means they're not really big molecules. So again, it's just kind of a simple way to allow transport of these specific hydrophilic smaller molecules through. So let's go back to that image. So phospholipids, that layer of the outer membrane, pretty straightforward phospholipids, and then the yellow hexagon, and then the yellow kind of rod-shaped thing. That's the lipoproteins anchoring in to the peptidoglycan. And then these pinkish cylinder look with that outer. And so there's different kinds of porin proteins that allow different molecules. So they're kind of specific, like this porin protein will allow this chemical. This porin protein allows a different chemical. So they're kind of specific. And they have them all, like they give them letter designations so that they know a fair bit now about all these different porin proteins. Okay, so then you have that anchoring to the glycan. And so this region where the peptidoglycan is located, that's called the periplasm. And that's, they don't really, you can't really show everything that's going on in that image. You get too busy. But the periplasm region is actually a very active area in the sense that there's a lot of enzymes and proteins, transport proteins, lots of stuff going on in here. And so it's the space between the inner surface of the outer membrane and the outer surface of the cell membrane. And it's only like 12 to 15 millimeters wide. And because there's so much stuff in there, it's pretty gel-like. Lots of proteins and enzymes. Lots of different flavors of different enzymes and proteins. And then the periplasm also has that really thin peptidoglycan. Okay, so very different. Gram-positive cell wall structure and gram-negative cell wall structure, very, very different. Here you're seeing the gram-negative electron microscope. So you can barely see that thin layer of peptidoglycan sandwiched in between the outer membrane and the cytoplasmic. Okay, so gram positives, gram negative. Tomorrow in lab, you'll try to pick out, you'll do that staining technique and you'll try to pick out which one's gram positive, gram negative. It's a bit of you have to be a little bit, it's a little bit of art and science when you do a gram stain. Those of you who've done med micro, you know what I mean. There's a couple of steps where it can go wrong. And so hopefully you'll get it, but don't feel bad if you don't get it the first time because it is a little bit of a challenging procedure. So that's why we devote one lab period just to the gram stain. Archaea, what about them? They have all kinds of different cell walls. It depends on where they evolved and adapted to, what kind of environment they're surrounded by. So they do have something that's called pseudo. What does pseudo mean when you hear that word, pseudo? Fake. Pseudo, so fake peptidoglycan. So what that means is archaea, by definition, they do not have true peptidoglycan. They have fake peptidoglycan. Very different. Because the way you define peptidoglycan has to have the NAM molecule, then it's true peptidoglycan. Archaea do not have NAM. They replace it with NAT. It's still a polysaccharide, you know, it's a sugar, hexose sugar, but it's a little bit different. So by the strict definition, archaea do not have peptidoglycan. They have fake peptidoglycan. So they have NAG-NAT, NAG-NAT, NAG-NAT. They have that kind of configuration. They also, because it's NAT instead of NAT, they don't have a beta 4. Instead, they have to do a beta 1,3 that bonds the NAG and the NAT together. So here's a NAG and NAT right next to each other, and you see the beta 1,3, and that one, your lysozyme in your saliva won't touch that. But that's okay, because archaea don't really bother us. They don't cause us problems. So they have pseudo, fake, they have that, but they can also have cell walls that are just sugar, polysaccharides, and they'll just stick with that. They could have cell walls that are mostly glycoprotein, so sugars and proteins bonded together in a particular pattern, or they might just have cell walls that's all protein. So because archaea can live in really different hostile environments, they've had to do these different cell walls in order to survive their particular habitats. Archaea and some bacteria as well can have an S layer. It seems to be more prevalent in archaea, but bacteria technically can also have S layer. Although a lot of the bacteria that you work with in lab, they've lost the S layer because they're lazy lab bacteria that get coddled and treated with everything they need, so they tend to pare down their cell wall a little bit. But this S layer, basically, it's just proteins put together in very particular configuration, and it can give some rigidity to the archaea. And actually, at least right now, the S layer wins. There's more archaea out there that use the S layer as their strategy to create a cell wall rather than. So the paracrystalline S layer can be quite successful in different habitats. And like I said, even like bacteria can do this too. Okay. So complicated. Let's do an eye clicker. Let's see if I can get this thing to work. Oh, my computer really does not. Like, all right, I'm gonna do it this way. Let's see if this works. Are you able to get in? Okay. Which of the following depicts the sugars? And you can look at your notes. See if this one's gonna work. More of a attendance participation point. So we're talking about domain bacteria, by the way. Domain, I have to put that in. Domain bacteria. Domain bacteria. Ten more seconds. Okay. So right now, can you see the poll? You can see it? See, it's not showing up on mine. What is with my laptop? So iClicker works for you all, but it does not. It's just bizarre. Like I have to do an extra step to see the poll. Like you can see it right now, the bar graph. I know, but you shouldn't see it until I hit results, and I haven't hit results yet. So it's like, anyways, I have to hit results. You already see it. So again, there's something bizarre. Anyways, neg nam, neg nam, neg nam, neg nam is correct, right? So um, answer A is correct. Okay, so I'll continue to work on the iClicker issue, but at least we can kind of get it to work. We're stumbling through it. All right. So I'm going to stop share so I can get out of this. Okay, so let me go into this view so that I can show you the cutoff point. All right, so for the exam, what I just threw at you is a lot. So today is Monday, the exam is Friday. So the content going through basically through slide 13 here, I want you to know the difference, like gram positives have a very thick layer of peptidoglycan, gram negatives don't. So the content that you see in slide 13 is fair game, but the details, like what is peptidoglycan in detail, I'm not going to ask. Okay? My reasoning for that is today's Monday. Learning the details of peptidoglycan is going to take you a while. And I don't want you to rush through that for Friday. So it's going to take a while for that to sink in. So we'll just do the big picture idea of what the difference, overall difference, cell wall of gram positives versus gram negatives. And then the slides 1 through 13, don't forget a lot of that is cell membrane material that we're talking about. So know that content as well. Okay, so draw the line after slide 13. That makes sense? I will post a study guide tonight. Give you a little bit more guidance and tips for the exam. And then remember, it's going to be 2 parts to the exam, the essay or the multiple choice, true, false, and then the essay. Okay. Okay. So I'm going to post also a second slide set that will start on Wednesday. Wednesday, we're going to get into, it's still cell structure, but we're going to look at some special things going on, like endospores, flagella, okay, but it's too big of a topic to get into right now. So we'll just stop right now. And we'll start that up on Wednesday.